my next speaker is Professor Harald Burkhardt from the Goethe Institute in Frankfurt am Main. And my name, as you heard, was Solbritt Rantapää Dahlqvist. I'm Professor of Rheumatology from Umeå, northern Sweden. Professor Harald Burkhardt, he started his academic education in Hannover and moved from there to Erlangen, Nürnberg. And he was appointed full professor at the Goethe Institute in 2006. And his research has involved several of the rheumatic diseases, but he has been focusing on rheumatoid arthritis and psoriatic arthritis, I believe. And he has been working with autoimmune mechanisms in the pathogenesis of collagen-induced arthritis in mice models, as well as in RA patients. And his research has been performed at the Goethe University, but also in collaboration with Fraunhofer Institute in Frankfurt. He will discuss today the spectrum of psoriasis and psoriatic arthropathy, I should say perhaps, involving both the innate and the adaptive immune system. And the title of his talk is, as you can see, Autoinflammation and Autoimmunity, Psoriasis, Skin and Joint Disease at the Midpoint of the Spectrum. So please, Professor Harald Burkhardt, the stage is yours.
Yeah, thank you very much for this nice introduction. And first I want to express my gratitude to the Royal Swedish Academy of Sciences for inviting me to honor Dr. Kastner on the occasion of the Crafoord Prize. And with the next slide, I will briefly show the disclosure of my potential conflicts of interest, because at the end I will show one slide that contains new medications, and as I'm a clinician, I'm also involved in clinical studies, therefore showing this. And first, congratulations to Dr. Kastner. He established the concept of autoinflammatory diseases, and that is the definition taken from the 2011 paper that represents the still up-to-date, generally accepted definition of the concept. However, for me it is important because I will talk not really in the immediate focus of autoinflammatory diseases, to say that I'm quite happy that his concept also included there is a continuum between the autoinflammatory and the autoimmune. And this concept was also taken up by colleagues like Dennis McGonigal and McDavid, who have formulated a kind of classification, a hypothesis on immune-mediated diseases, and this depicts a graphical presentation where the distinct disease entities are arranged according to the assumed contribution by innate and adaptive immune mechanisms to their pathogenesis on a continuum between two ends that are represented at the top by the monogenic autoimmune disorders like the Mediterranean fever, and at the very bottom, very rare autoimmune diseases caused by mutations in, for example, transcription factors like FOXP3 or the autoimmune regulator AIR. And the psoriatic diseases, so skin disease as well as joint disease, have been placed into the category of intermediate pattern diseases, and therefore the title of my talk. And however, I'm not a dermatologist, and therefore I'm a rheumatologist, so I will focus at the midpoint of the spectrum on psoriatic arthritis. And this is a disease that has a very broad spectrum of clinical manifestations. People that develop the first symptoms have already, usually in 80% of the cases, already psoriatic disease, and very rarely the musculoskeletal manifestation precedes. Up to 25% of the patients with psoriasis after having the diagnosis of the skin disease will develop within the next 25 to 30 years also a psoriatic arthritis. And the disease can manifest as a peripheral joint disease, as you can see here, but also as a spondyloarthritis of the exoskeleton. It can affect para-articular structures like the anatomical site of the insertion of the tendon to the bone, the so-called enthesis, that is enthesitis, or can manifest as inflammation of a whole digit in the hand, or the toe, which is called dactylitis, and we have also extra-articular manifestations. Now, the disease is a complex disease. It is, has environmental or lifestyle factors. One lifestyle factor I have to mention is physically demanding occupational tasks with heavy weightlifting, and on the other hand, and I will focus on that because it was in the focus of my interest, genetic factors with common loci with skin disease, but also psoriatic arthritis-specific loci. The next slide shows somehow the jigsaw of psoriatic arthritis genetics. And there are different loci that are then somehow arranged into certain pathways. It's important that many risk loci arrange into the IL-23, IL-17 pathway and NF-kappa-B signaling, and then in mechanisms of adaptive and innate immunity. However, there are also, of course, an MHC class 1 association with a major risk factor for psoriasis in the C allele 0602, but the psoriatic arthritis is associated mainly with B alleles, especially B27. And the question is, how do these different risk alleles contribute to the susceptibility for psoriatic arthritis? And that shows that immune cells are usually selected for stimuli response characteristics that operate within a certain range of values that, however, might differ in between individuals due to the underlying genetic or epigenetic factors. And therefore, such a response curve from one individual can be shifted, for example, to the right-hand side due to underlying variations in the underlying genes, probably leading to a loss of function mutations, increasing the threshold for activation. But it can also be, of course, the other way around. Thus, this individual variation of the threshold of activation towards a wide array of immune stimuli into responses might affect the susceptibility or resistance to immune-mediated diseases. So one example is given on this slide from our own association studies. It shows the genetic association study for quite a lot of different single nucleotide polymorphisms, and on the y-axis is given the p-value for association, and here on chromosome 1 one can see there's a single peak of major significance that is genome-wide, and this is in a gene which is called PTBN22. It's a phosphatase that plays a role in the T-cell receptor signaling, and it's a coding variant leading to replacement of an arginine by tryptophan. And the role of PTBN22 is somehow to have an inhibitory effect also on T-cell activation, and it is not clear how to say really to pinpoint to the effect of this single mutation. It is just known because the respective functional data is inconsistent, but what can be shown is that this is associated with a number of autoimmune diseases, especially those that have also a kind of B-cell signature. That means there are autoantibodies that are used for the diagnosis of these diseases. For example, rheumatoid arthritis, the production of autoantibodies directed to citrullinated, that means post-translationally modified antigens. In psoriatic arthritis, we do not have such diagnostically used autoantibodies. However, we have investigated for these APGA antibodies, also patients with psoriatic arthritis, in a very large cohort of patients that was well characterized. And we could detect, however, in a subgroup of patients that they produce autoantibody to citrullinated peptides. And those patients have been shown to have a more severe disease. It is correlated with erosive disease, and this shows, at least in a subgroup of those patients having peripheral arthritis, obviously autoimmune reactions could contribute to the clinical phenotype. In addition, we have also signs in patients with psoriatic arthritis of the activation of the adaptive immune system. They're activated T cells that are differentiated in a pro-inflammatory phenotype, producing interleukins like interleukin 17, IL-22, IL-21, TNF-alpha, GM-CSF, both in the CD4 as well as in the CD8 compartment. Now I have to mention the dominating subphenotype are the CD8 positive T cells in the synovial compartment, and they have been shown to also clonally expand and be associated with erosive disease, which is consistent with the MHC class 1 association, and especially the B alleles. Now I will come for a moment to HLA-B27. It has a quite conventional role in presenting peptides to the respective CD8 positive T cells, peptides that are derived from their own peptide protein synthesis. However, HLA-B27 is very special. It has a cysteine residue that can lead to the formation of a aberrant MHC class 1 molecule in form of a heavy chain dimer. And this is also presented at the surface of the cell. It's unstable and can then activate a subset of CD4 positive T cells that express a subtype of a killer cell immunoglobulin-like receptor to activate it. And on the other hand, this homodimer leads to the initiation of an unfolded protein response, because this homodimer molecule has problems with conformation. And accordingly, as a result of this unfolded protein response, interleukin 23 can be produced. And this is a differentiating factor of the pathogenic phenotypes of the Th4 and of the Th17 and TC17 T cells. So I have shown that probably IL-23 has a critical role in the pathogenesis for driving these pathogenic T cell subpopulations, and we could see in our genetic studies, in the association studies, that also this pathway is associated with allelic variants, but not with the IL-23 gene itself, but with the IL-23 receptor. And this shows the association plots. And in the upper plot, you can see a signal by the red dots with 13 credible SNPs for psoriatic arthritis, and this could be localized to map into a region of promoter and enhancer regions in CD8 positive memory T cells, and based on available public domain data from respective epigenetic studies. This signal was different from patients with psoriasis arthritis. They also show association with variants in the IL-23 receptor gene. However, this is localized different. And interestingly, one of those SNPs is identical with an earlier coding variant that has been described in association with inflammatory bowel diseases and has also been shown to really lead to a higher upregulation of the receptor and an improved signaling. So this pinpoints to a potential role also of mucosal immunity, both in also in psoriasis or psoriatic arthritis, and indeed there are endoscopical studies in patients with psoriasis arthritis that shows that more than 50% of the patients have a subclinical enterocolitis. And interestingly, also these patients have a dysbiosis in the intestinal tract. And to this, also the HLA-B27 is a contributing factor, because it has been shown that B27 can lead to a reduced species enrichment in the intestinal microbiome, with the reduction especially of Akkermansia and Romanococci. So the IL-23 has in this respect a complex role. IL-23 has both homeostatic but also pathogenic effects in the intestinal tract. And it is therefore possible that during chronic inflammation, not only there occurs a local upregulation of IL-23, but this cytokine may also increase in levels in the general population, in the general circulation. And a couple of years ago, there had been a very elegant and intriguing cellular connection being described between the upregulation of the IL-23 in the circulation and the onset of pathology at the enthesial localization. And this was a group of Sherlock et al. And what they did, they upregulated IL-23 in an animal model by using DNA microcircuit technology. And what they could show that they have a responding T-cell population localized at the enthesis that are CD4, CD8 negative, expressing Rho gamma T, and get activated by IL-23 to produce other pro-inflammatory cytokines like TNF-alpha, IL-17, and IL-22. And these then cause the changes in the tissue, inflammation, bone erosion, and osteoproliferation. So the population of these cells is also present at the enthesis of the axial skeleton, and therefore contributing also to spondyloarthritis. And for the human system, these population of T cells have been identified as gamma delta T cells and ILC3 cells. So these are classically cells that belong to the innate immune system. In addition, we have to think that entheses are sites of exposure to mechanical load, and usually ligaments and tendons, of course, they require for the maintenance of their architecture, physiological load, mechanical load. However, if there is excessive mechanical stress, this leads to the initiation of immune-mediated inflammatory responses. And looking at the enthesis, there is a very close anatomical association between the structures like the tendons and the fibrocartilage and the synovial component containing macrophages that can respond vigorously to occurring damage in the connective tissue. So fragments of the connective tissue can function as so-called danger-associated molecular patterns to activate the local stromal cells via the respective innate immune receptors and the macrophages. And in addition, what plays a role for the pathogenesis of enthesitis is that via the circulation, also microbial adjuvant can reach these enthesial compartment from distant sites of the leaky gut. And these microbial components can activate locally then also the toll-like receptor to amplify this inflammatory process in a kind of gut enthesial connection. So I will come back now to our very early studies on genome-wide association studies and back to the IL-23, IL-17 pathway. And this shows the results of a genome-wide association study in a heavily underpowered population of psoriatic arthritis patients, which we performed in Germany. However, what you can see and therefore I'm showing it with the red circle, so somehow surprisingly, we found a genome-wide already significant signal in the TRAF3 IP2 gene. And that's a very interesting gene because this encodes for the E3 ubiquitin ligase ACT1. And this is an important molecule as shown on the lower panel that has a role in connecting the cytoplasmic part of the IL-17 receptor towards the NF-kappa B signaling pathway by interacting with TRAF6. And in a replication study in a European collaboration on the European Psoriatic Arthritis Consortium, we could replicate our study and we could identify by exon sequencing a coding variant where there is an exchange of the aspartic acid in position 10 of this molecule by an asparagine. And we were then interested on the functional consequences and performed mammalian 2-hybrid luciferase reporter assays, and we could replicate what was known, that TOF6 binds very nicely in this assay to the wild type ACT1. However, when we replaced the glutamic acid by an asparagine, this interaction was completely destroyed. This means we have a loss of function mutation, and this is surprising. It is counterintuitive to have a pro-inflammatory cytokine, which plays obviously a central role also for psoriasis, which is known now, and to have this associated as a risk factor with psoriatic arthritis. And this surprise also stimulated other groups to investigate it. And especially the group of Xiaoxiao Li at the Immunology and Lerner Institute in Philadelphia, and they generated ACT1 knockout mice or deficient mice. And surprisingly, what these mice have is an highly activated, hyperactivated Th17 population. So these T cells produce a lot of IL-17 and IL-22, although the signaling pathway for IL-17 is somehow compromised. These mice develop, because of the IL-22 production, a spontaneous skin disease that is IL-22 dependent. However, the T cells that have this Th17 differentiation, they require a signal via the IL-23 receptor to differentiate into this phenotype and to stabilize it. And what this group could show, and this was very interesting, that ACT1 does also have a role in the IL-23 signaling, but in a quite unexpected way, because ACT1 can also bind to the transcription factor STAT3, which plays a role in the transmission of the IL-23 signal, and by binding it has an inhibitory function on this pathway. And then, of course, if we have a deficiency of this factor, STAT3 is upregulated, and accordingly also an overproduction of the pro-inflammatory cytokines. And now comes it also if we take, and this is what they showed, when they introduce this mutation, it has the same effect as a total deficiency, and this explains why this variant might be associated with psoriatic arthritis, because it somehow leads to a hyperreactive Th17 and probably also TC17 phenotype. So there is another aspect, because I told you that this mutation somehow compromises the IL-17 pathway, and IL-17 is on the other hand also a factor that is plays a role in the host defense against candidiasis. The question is, was then also, could this also somehow contribute to the pathogenesis? And first, there is an increased prevalence of mucocutaneous candidiasis in patients with psoriasis. However, this could just be an epiphenomenon and not necessarily the cause of psoriasis. However, please just keep in mind that it might be that, or it argues that at least there is a higher likelihood for exposure to fungal, antigenic, or structural components. And this is important for introducing data which relates to an animal model that has been established in the lab of my dear friend Richard Holmner, with whom I'm collaborating a lot. And he could induce an animal model of psoriasis and psoriatic arthritis by the intraperitoneal injection of Manan from Saccharomyces cerevisiae. And as you can see, that's a perfect phenocopy of psoriasis. And when you look at the digits of the paw. It's a typical dactylitis that we know from patients, as well as the end joint involvement. And this is an animal model that is independent of alpha beta T cells or B cells, so it does not need the adaptive immune system. However, it requires granulocytes, and it can be blocked by antibodies to IL-17A. What is also shown, and that's interesting, if the mice are made deficient of the production of reactive oxygen radicals by NCSF deficiency, those mice have a much higher, have a much more severe disease with higher scores of arthritis as well as higher psoriasis scores. Moreover, it was then also investigated what is the role of nitric oxide metabolites that are produced as a consequence of the activity of nitric oxide synthase, NOS2. And these metabolites have been shown to be increased not only in the periphery of the serum, but also in the peripheral tissues, so within the skin and in the joints, shown by luminescence imaging of peroxynitrite. And when now the NOS deficiency was investigated in an NCSF deficient background and looked what's the disease severity that develops, then it's clearly shown that mice that are made deficient of NOS2 have a reduced severity of this disease, indicating in this term that by contrast to reactive oxygen radicals, the NOS-derived metabolites are disease promoting. And additional studies then showed that in a NOS-dependent manner, the production of interleukin-1 alpha by skin macrophages is critical for arthritis development by leading to an increased production of IL-17A by innate lymphocytes. This shows quite complex, as I think, mechanisms of how Manan can induce an innate immune response leading to psoriasis and psoriatic arthritis. To show the data in the humans, there is an association of NOS2 with psoriasis as well as psoriatic arthritis. However, it is very often in polygenic disease, the functional consequences of the respective sequence variations are unclear. However, what we could show in humans is that we could detect an increased upregulation of NOS2 in a subset of mononuclear cells that were challenged with LPS and interferon gamma in the peripheral blood from patients with psoriatic arthritis compared to healthy donors. And this indicates that probably this pathogenic effect of nitric oxide shown in this animal model might also be relevant to the human disease. And now I'm coming to my last slide to summarize. So I've shown you adaptive mechanisms that might contribute to psoriasis. However, only in a subset of patients clearly, and probably mainly in peripheral arthritis, whereas innate immune mechanisms dominate in enthesitis. And there's a link in between both by the IL-23, IL-17 pathway. And this has also been turned out as a fruitful target for development of new drugs, both in psoriasis and psoriatic arthritis. We have at least 4 antibodies targeting IL-23. Now also JAK inhibitors that target the respective signaling pathways, and at least 3 antibodies directed to interleukin-17 and 1 to the interleukin-17 receptor. And with the last slide, I thank you for your attention, and I have to thank my collaborators, especially Rickett and his group, but also our collaborators from the European Psoriatic Arthritis Consortium, and last but not least, my collaborators and my, my team at the University in Frankfurt. Thank you for your attention.